Okay, now we're going to talk about the body fluids and the circulation. What can you tell me? What are the body fluids? Something inside the body, the blood, the connective tissue that connects all the organs, right? So, can I say that blood is a major connective tissue in the body? Okay. Apart from that, we do have some body fluids. Yes, like lymph, devoid of RBC, is called as a lymph. Whereas RBC is not there, lymph will be there. Apart from that, we do you have other body fluids that we are going to study in this chapter. So let's start. So body fluids are the medium of transport. So what they are actually? Medium of transport. What do you mean by transport? In case of blood, what is happening? Tell me. Exchange of the gases, that is oxygen and carbon dioxide, exchange of the food, and removal of waste will be there. When we take breath, what is going to happen? We take a air. We are not taking oxygen. We are taking whole air. Whole air goes to the alveoli. There is a blood vessel connection is there. From there, it goes to the hemoglobin. So hemoglobin accept the oxygen and relieves the carbon dioxide. Okay. So you can see that there are two things are there, right? So one is taking air, other is taking air outside, which is little bit warmer because the body temperature is there, right? Yes or no? From body that's coming out. So once the oxygen goes into the blood, what is going to happen? It bind to the hemoglobin. Now hemoglobin is going to travel all over the body. Which cell is required? It to give to hemoglobin to that, and again carbon dioxide will be taken, and the waste will be directly into the blood. This part is clear. Okay. So body fluids are the medium of transport of nutrients. You got the point? Then oxygen along with carbon dioxide also there, and other important substance like glucose, right? That will be there. Now body fluids. Achha, before we go ahead, I want to talk about this. What is the active transport and passive transport? The active transport is the one who requires the energy. And passive means what? If I'm moving, along with that, once I open the door, you also moving. So I expenditure my energy on the door, opening of the door. So did that? No, you just go. So similarly, if a glucose is going, for example, some other particles also going along with the glucose. That is passive transport. Energy expenditure is not there. Okay. So let's talk about the body fluids. There are some intracellular fluid. What is intra? I told you intra, within, inside, and inter. No, intra means actually within the. See, this is a cell. This is a cell. This is intra. Inter between this and extra outer. So, fluid of each cell, cytoplasm will be there counted. Extracellular fluid, blood, lymph, cerebrospinal fluid. What is cerebrospinal fluid? In the spinal cord or that cerebral. What is that? Cerebral fluid is there. For what? Shock absorber. Very good. Because shock absorber has to be there. Very good. So, you know everything. So, blood is the most commonly used body fluid in a most of the higher organism. Lymph is also transports certain substances like protein and fats, but not RBC. Getting my point? RBC is not there in a blood. If it is devoid of the RBC, that is called lymph, and lymph is responsible for protein and fat. Why? In cerebrospinal fluid, or that is the fluid which is responsible for a lymph only, right? Lymph is what? Where RBCs are not required, so lymph will be there. So obviously it's going to transport protein and fat because cells are there, right? Yes. Now blood. The blood is a fluid connected tissue composed of the fluid matrix, plasma, and the blood corpuscles. It forms 30 to 35% of the extracellular fluid. So out of extracellular fluid, blood is how much? 35%. 30 to 35%. It's slightly alkaline, having the pH 7.4 or 7.35, also written somewhere in books. Okay. So this is the pH of blood. Now, blood is composed of how many things? Let's see. First, form element. Form element, erythrocyte. Erythrocyte is what? Your RBC. Leukocytes, thrombocyte. Thrombocytes, your platelets. Okay. Now, in plasma, there is a water, protein, and mineral. Can I say that blood is major component of the plasma? And inside the plasma, there is a form element. I can say that if you take a blood of someone's like patient is there, you just take out the blood and you keep in the test tube. After some time, you'll get a straw yellow liquid color. Suppose this is a test tube. Okay. Here, you get a straw yellow color, which is a plasma, and the red color will be like this. So what do you mean by that? Plasma is major amount and this form element are suspended inside the plasma. What do you mean by suspended? They can move in the plasma. This point is very clear. Okay, what is the function of RBC? To carry out the exchange of gases. What is the function of WBC? Phagocytosis, that is fight against the infection immunity. What is the function of thrombocytes? That is platelets, blood clotting will be there. Okay, vitamin K is required in the thrombocytes. Okay, and what is the function of plasma? To give the viscosity to the blood. Otherwise, form element are not able to transfer, right? Getting my point? So plasma is required for that. This point is clear. Now move to the this point. Now plasma is a straw colored viscous. Just I told you, right? What is plasma? It is a straw colored viscous. What do you mean by viscous? Who give the viscosity? What is viscosity? I have a oil in my hand and one bottle of oil. Sorry, one bottle of oil, one bottle of water. I just spill it off. Who goes first? Water. But oil take time. So can I say that oil is more viscous? Viscous means what? It has a fluidity in a viscosity manner. Okay. So viscous fluid means what? It's not able to flow directly. It's not able to spread directly. It takes little time. So it consider 55% of the blood volume. 45% is form element. 55% is what? Plasma will be there. It consists of 90 to 90% of water. That's why water is needed, right? If you feel dehydrated, so we'll feel some sickness will be there. Why? Because water has to be there. If there is a blood loss, like some person come met with the accident, hemorrhage is there. What is hemorrhage? More blood loss will be there. So they are not into the consciousness because blood is a connected issue. It required for circulation. If it is not there, what is going to happen? See, if you have a small cut, it will be okay because it will be clotting properly. Time itna nahi lagega. But in case of high hemorrhage, accident, what going to happen? A major blood loss will be there, and the person is under consciousness. Okay, so tell me one thing: 6 to 8% protein. What kind of protein? Fibrinogens. Fibrinogens is a fiber-like structure required for clotting. So vitamin K along with the fibrinogens required for a clotting in the case of platelets. Okay. Then albumin and globulins are the immunological proteins are there. They play important role in the immunity. Then glucose, obviously for energy purpose, amino acids for the synthesis, and small amount of minerals like sodium, calcium, chloride. Now tell me one thing. When we are sick or when we are our diarrhea or when we are feeling not much good, so we drink electrolytes. What is electrolyte? Like salt water and little bit sugar will be there. Even in if you go to the pharmacy, you will get that electrolytes ready made. Those who are suffering from the diarrhea or a sickness, they will get it that. Why? Because in the blood, sodium is there. In that electrolyte, what is that? Sodium, calcium, and chlorine ions are there. Now, for relaxation and contraction, what is required? We just did yesterday. Some channels are required, right? So myosin is required for contraction, but inside the myosin, that is cell, sodium and potassium. When sodium go out and potassium come out, relaxation will be there. When potassium come out and that vice versa, contraction will be there. So we need a what? Ions. So are you getting my point? Why we feel sick? Because these ions are not much up to mark. They are removed by the urination or the excretion process. So we need iron. For that doctor prescribe or just we can feel we are feeling sickness, so we can have this. Okay, but you can't eat direct salt. It is problematic. More amount of salt will be leading to problem. What? More ions will be there, and there will be a problem will be there, right? So mostly diabetic person don't eat that much salt. Okay. Now erythrocytes, leukocyte, platelets are collectively called a form element. So what are the form elements? Erythrocyte, leukocyte. Erythro RBC, leukocytes your WBCs, and platelets your thrombocytes. Erythrocytes are the most abundant cell. Erythrocytes are what? Most abundant. That's why blood is mostly red in color. Cells in a human body. Total blood count of RBC is 5 to 5.5 millions, which is slightly less in the female due to the menstruation. So in the male, you find 5.5 millions of the cell in a adult male. But in case of female, because of the menstruation, some RBC are removed off. So they have less. So mostly in case of female, they have a chances of anemia. Okay. Now, what happens if the anemia is there? What is that cause? RBC is not that much mature, or RBC is not that much capable to carry the oxygen. So in the body, there will be a less oxygen. If there is a less oxygen, obviously respiration process is not up to mark. So energy production will be less, and the anemic person is lethargic. I mean, like, they're not able to do the things very excitement, okay, or with energy. So it form in the bone marrow. So where the site of erythrocyte? In the bone marrow. So erythrocyte form in what? In where? Bone marrow. Site of erythropoiesis. Formation of erythrocyte is called erythropoiesis, which is formed in under the bone marrow. Point should be clear, okay? So tell me one thing: if the bone is not proper or bone is fractured, you may contain the less. You may contain the less RBC. It will be a problematic. So nucleus is absent in a mammalian RBC having the biconvex shape. Can anybody tell me why this is so? Why nucleus is absent in a RBC? What is the function of RBC? To carry the maximum, yes, maximum capacity should be utilized. If there is a nucleus inside the cell, what is going to happen? It's going to cover little area. If there is no nucleus, a maximum amount of energy, maximum amount of space can be utilized. Maximum amount of space can be utilized for what? Oxygen or exchange of gases, we can say. So because of this, RBC is lack of nucleus. Initially, when the RBC just form it with the help of erythropoiesis, they have a nucleus. But once they mature, nucleus is not there. What is the reason? Maximum amount of space should be utilized for the exchange of gases. This point is clear. This asks for 1 mark. Okay. Now, every 100 ml. Now, every 100 ml of blood contain 12 to 16 gram of hemoglobin. How much hemoglobin is there? 12 to 16 gram in 100 ml. They have a lifespan 120 days. They are destroyed in a spleen, or we can say liver as well there. So spleen is called as a graveyard of RBC. Along with there is some function of liver is also there. Now, leukocyte. What is leukocyte? WBC. Leuko means what? White color cells. Okay. So WBC are a colorless due to the absence of hemoglobin. There is no hemoglobin needed because they are into the phagocytosis. Immunology system is there. So how many there are? 600 to 800 of the WBC are present in each ml of a blood. But in case of male and female, only difference is RBC. Male will have a more RBC, female will have less RBC because of the menstruation. This point is clear. Now, we're going to talk about the next one, that is WBC have a two types. Granulocyte, that means on the surface, the granules are there. And agranulocyte, that means the granules are not there. For like reticular smooth ER or rough ER. Ribosomes are present, that is called rough ER. That is undergo the protein synthesis more. And when you talk about the smooth ER, that means what? They do not contain the ribosomes will be there. Similarly, agranulocytes are the one who contains the granules, and agranules are do not contain any granules. First, eosinophils, basophil, and neutrophils. Agranulocyte, lymphocytes, and monocyte will be participate in that. So what they are, let's study about it. Okay. So when you talk about the neutrophil, can you see the diagram? This eosinophil, basophil, neutrophil. I will tell you why they are there. See, to observe something under the microscope, we have to perform the addition of dye. Okay. So this take up eosin dye. These take up alkaline dye, and this take up no any dye. I mean, both dye can be worked here. So eosinophil, basophils, and neutrophils, they are on the basis of which dye they take up. Getting my point? And this monocytes and B lymphocyte we're going to talk about. So neutrophils are the most abundant and the basophils are the least abundant. So basophils are very less, neutrophils are very high in quantity. WBCs, monocytes and neutrophils are phagocytic. So monocyte, can you see the size? Monocyte and neutrophils are the part of phagocytotic department. What is that phagocytotic? If there is any bacteria is there, harmful bacteria pathogens, they will eat up, and whatever the remaining part of that bacteria is there, they will show on the cell wall. Why they do this? Because when they pass from the bloodstream, every neutrophil, every monocyte should understand this is the foreign particles. If they come again, just kill them. Okay, so that is a phagocytotic cell. So phagocytotic cell have a two functions. First, to kill it. Second, whatever the remaining part or a protein part of the antigen should be stick on the cell wall of the this monocyte or neutrophil to indicate if the again same protein comes, you have to fight it against. Getting my point? Okay. Now, which destroy the foreign organs? Basophils secrete the histamine. Now, tell me one thing. What is allergy because of the histamine? We take a cetirizine from pharmacy. You take a cetirizine. If you get an allergy from something, you get a medicine known as cetirizine. It is H1 receptor blocker. It is histamine blocker. If we take a medicine, histamine in the body will never secrete it. Histamine we getting from whom? Basophil. Basophil like alarming. Basophil. If there is a problem in the body, suppose any foreign particle is in the body, will start sneezing. Because of what? Basophil act like a alarming. Okay. So basophil and serotonin and heparin that are involved in the inflammatory reaction. So what is the inflammatory reaction? Tell me one thing. Suppose I get a harm on here, so it will be get swollen, it will be reddish. Yes or no? Why? Because there will be the inflammation. But there is a systemic inflammation. What is systemic inflammation? Inside the blood, there is the inflammation because of the foreign particle. So who will responsible for that? Heparin and serotonin involved in an inflammatory reaction. Now, serotonin is a good hormone, right? Yes, correct. But it is involved in a inflammation because more excitation is there. Cell undergo the inflammation. Now, eosinophil resist infection and allergic reaction. B and T lymphocytes are. What is B and T lymphocyte? T lymphocytes is the memory one. B lymphocytes are responsible for formation of. Last time we have learned something. Antigen, antibody complex. Okay. So T are memory cells. T cell will be what? Now tell me one thing. Monocytes have what done? What they have done? They kill the pathogen and they spread the protein on the cell wall. Now these T lymphocytes, along with that, they will collect the data that this protein has been arrested. That if we come again or this particles come again, then kill them. So this information is stored in whom? T lymphocytes. Now T lymphocytes and B lymphocytes are responsible for synthesis of antibody complex. So T lymphocytes are required. The antibody are secreted by B lymphocyte. What is antibody? Against the antigen. Antigen means what? Of harmful bacteria or pathogen will be there that has to be killed. That has to be locked. So antibody will be secreted. Who will secrete antibody? Beta lymphocyte, B lymphocyte. But for beta lymphocytes to secrete a specific antibody, definitely they require T lymphocytes as a memory. Now tell me one thing. ELISA test is there. So what is that? How I come to know that I have an infection in body? I do the blood test. In blood test, what they do? They don't do the virus test. They do the antibody test. So in the blood, if there is an antibody is found, according to that antibody, they can find which disease or disorder is there. Getting my point? So we can't check. We are going opposite. We find the antibody. If there is an antibody, definitely there is an antigen. Yes or no? So every antigen, for every antigen, there is a specific antibody is produced in the body. For example, everybody has a taken corona vaccine. So in our body right now, there is an antibody for specifically for corona one. Okay, they will get activated when we get infected. Getting my point? Is it happen once you got corona, for example, minute, everyone has the exposure, minute exposure. What actually happened? Your body is secreting the antibodies. So inside the body, there is information already stored. This point is clear. So who is phagocytotic cell? Monocytes and neutrophils. But along with that, beta lymphocyte, B lymphocyte, and T lymphocytes also responsible. Okay. This point is clear. Let's go further. Now we're talking about the thrombocyte. What is thrombocytes? Platelets. What is platelets? They are the fiber-like structures. Fibrinogen from the plasma and vitamin K is responsible for formation of clot. If suppose I get a cut over here, after few seconds, blood will be oozing out. But after few seconds, there will be a all. Suppose this is the cut, okay, from the body. Now this is blood is oozing out. What is going to happen? This thrombocyte come to know that there is something opening because a lot of thrombocytes are there in the blood. RBC and thrombocytes are very, very much in larger size. So what going to happen? They start the seizing this thing. Okay, this are the blockage of the opening. Now you can see here, if I have, okay, can you see the dark brown color over here? What is that? It's a clot. It's a clot. So blood is what red color mostly. But when thrombocyte makes it and the fibrinogen and vitamin K is there, the clotting will be there. And this is what, if I do scratch on this, I will get again blood. Why? That's a dried part. Until unless there is no skin forming, I can't do this. Okay, but by mistakenly sometimes we do, right? Yes. So that is the mechanism. Now tell me one thing: if vitamin K and thrombocytes are not there, what's going to happen in that patient? Coagulation will be not much faster. So more blood can be lost, more hemorrhage will be there. So it is very problematic to that person. Okay, now thrombocytes are a fragments. Why they are fragments? Cell fragments. They do not have a nucleus at all. These are the fragment-like structure. कपड़ा फटा है तो उसको हम क्या करेंगे? What will do? Snitching, right? Snitch करेंगे ना उसको? Stitch करेंगे, right? तो क्या क्या? For example, this is a part. अभी यहाँ पे what we'll do in this case, I will do this threading like structure and then we'll close it up, right? Similarly, thrombocytes are responsible for the cell fragments produced from the megakaryocytes in a bone marrow. From the bone marrow, we get erythrocytes along with megakaryocytes. This megakaryocytes further develop into the thrombocytes. This point is very clear. Okay, how many? How many? 1,50,000 to 3,50,000. Why they needed? Yes, they needed because body don't know now how many cuts will be there, how much area will be affected, right? So they each ml of the blood. Platelets are involved in the clotting or a coagulation of the blood in case of injuries. Blood group. What are the blood groups? Can anybody tell me about the blood groups? What is a blood group? Tell me. Okay, when you talk about the blood group is positive or negative. Positive or negative, it depends on the RH, positive RH. Rhesus monkeys. On the RBC, if there is a RH factor, rhesus monkeys factor, we are originate from Homo sapiens. So some are transferred, genes are some transferred. So on the RBC, if there is a RH factor, rhesus factor is there, it considers a positive. If rhesus factor is not there, it considers as a negative. So positive and negative will be there. Tell me. Now we're going to talk about the blood groups, and blood groups are very interesting one to understand. If we have a antigen A on the body or A on the RBC, blood group will be the A. Antigen B, blood group will be B. Antigen A, B, and both are there, blood group will be AB. And if your parents are not loving you, means what? There is no antigen at all. So it's zero. That is O. You can't say na zero. O. So what is that? O is the donor. Why it's a donor? Because it do not have any antigen. So there will be no resistance if the blood is transfused from the person having the O positive to the person. But can I, if I'm O positive, can I take from blood group A, B, AB? I can't take it because my body will be reflected back because I need only O positive then O positive. O negative is the very rarest one. So what is O? Zero, no antigen at all. So they act like a donor. Okay. And who is act like a receiver? Yes. So blood groups are there. Antigen. What is antigen? Antigen. A foreign particle is present on the RBC. So if the antigen A is present, well, blood group is called A. So they are anti-B. Means what? They can't take the blood B. Now we talk about the blood groups. So when we talk about the blood group A, it has the antigen A. B, antigen B. AB, both antigen A. O, no one loving the O because they are nil antigens, right? So donors group is mostly what? O can be donated to everyone, and AB is the who is receiving. Yes or no? They can receive us. Okay, this point is very clear. Now, I told you the RH positive, RH negative. Okay, we're going to discuss that. During the blood transfusion, what is transfusion? Is it possible just transfer blood like this? No. We have to take a test. Suppose a person is suffering from the AIDS, syphilis, or something, and we directly transfer that blood. The problem will be the patient. Okay, even the alcohol drinker is not allowed to transfer the blood. You can detect, you can take a test. Okay, now blood of the donor has to be matched with the blood recipient. Avoid the clumping of the RBC. If the clumping of the RBC will be there, what is clumping? They form a clot. If the blood is refused inside the body after just transferring, clump of the RBC will be there because RH positive, RH negative. That see the second baby I talk about now how will died because of the clumping or RBC. Now universal donor is O, universal recipient is AB. Now let's talk about this RH grouping. This is very interesting one. RH antigen similar to the rhesus monkeys are observed on the surface of the RBC. This is RBC, surface of the RBC, some antigens are there. Rhesus monkeys is a satisfactory move from my side. So on the rhesus monkey, that is RH factor is present if the on the surface of the RBC, that is RH positive. If there is no RH negative, RH negative are very less. So majority of the individuals, about 80%, such people are called the RH positive. 80% are RH positive, and those in whom antigen is absent called RH negative. Very less are there. Now erythropoiesis fetalis. What is that? Erythroplastocysis. What is fetalis? Fetus is not developing properly. So let's see. If I just show you this case, right? I just discussed this. If the father blood is RH positive and mother blood is RH negative, the fetus blood with RH positive. Why? Dominant is one RH positive right now. During the delivery of the first child, there is a possibility of exposure of mother blood with the fetus blood to develop antibodies. During the first delivery, what is going to happen? See, obviously, exchange one sperm, one ova, when combined, then definitely chromosomal exchange information will be there. So, at the first stage of zygote, they understood that blood group is RH positive, RH negative. Let's go with the RH positive because that is the dominant one. But mother body, because where is the fetus is going to be there in the mother womb? Definitely inside the mother womb. Mother body is going to exposure with the placenta. So obviously, in case of that, mother is RH negative. In this case, when a zygote is developing into the fetus cells will be going to have a communication between the mother cells. So mother blood with the fetus blood to develop the antibodies. So whenever the fetus is there with the RH positive inside the mother womb, mother will be developed into the antibodies. On first baby, they are not going to active. They are not going to act on the first baby because already zygote has been formed. Okay. In a mother blood, in subsequent pregnancy, the next one, the mother's blood can leak into the fetus blood and destroy the fetus RBC. So they can there will be a lumping of RBC will be done. Once there is a lumping of RBC inside the fetus, what going to happen? Obviously, RBC is not their body is not going to perform. So fetus will be die inside. That is called erythroblastosis fetalis. This point is clear. Again, so RH grouping when you talk about RH grouping when you talk about, we talk about the RH positive, RH negative factor. Now we have talk about the erythroblastosis fetalis will be there. Mostly this combination is also taken care. Okay, let's go further. And now we're going to talk about the case is called the erythroblastosis. Why this is going to happen? Injured tissue will be there. Once the tissue is injured, definitely there is an exchange of the blood will be there. So release of thromboplastin along with calcium protein and prothrombinase. Nase means ASE enzyme will be there. So this prothrombinase is converted to prothrombin to the and fibrinogens are converted. See what we are talking about. We're talking about the clot. How the clot is forming. If there is an injury, there will be a blood loss. So in the blood, thromboplastin, calcium, and prothrombinase get activated. Get what? Activated. Once they are activated, the thrombinase, ASE, is the enzyme is going to trigger the prothrombin. Pro means what? Pro means the it's not a thrombin, it's a prothrombin, the earlier version of the thrombin. So the prothrombin and fibrinogen are activated right now. What is fibrinogen? Fibrinogen triggers the fibrin molecules, and fibrin along with the blood cells form the clot. Are you getting my point? This is a cascade mechanism. So who is get activated first? Thromboplastin, calcium, and prothrombinase. Prothrombinase is going to talk about the prothrombin activation. Then fibrinogen activate. Then they activate the fibrin, and fibrin along with the blood cell forms the clot. Okay. We will see the video of the clotting. Okay. Just 1 second before that. When an injury is caused to a blood vessel, bleeding start, which is stopped by a process called a blood clotting. This is definition. You should write. I mean, like in examination, they ask. An injury or a trauma stimulates the platelets in the blood to release the certain factors. There are many 12 factors are there. From 12 factors, like factor number 1, factor number 2, factor number 3. We don't know that. Scientists has given them 1, 2, 3 till 12 name that activate the mechanism of coagulation. Calcium play important role in the blood clotting. So calcium has to be there. Now let's learn about the clotting mechanism. If there is an injured tissue, what do you mean by injured tissue? Injured tissue will be there. If there is an opening on the skin, blood will be oozing out. Okay, now injured tissue is going to underward. Injured blood vessels as well. When there is an injured tissue, obviously blood vessels are injured. That means blood vessels are open right now. So collagen fibers in vessel wall exposed. So if the wall got break off, if the wall undergo the break off, what going to happen? A collagen fibers, adipose tissues, collagen fiber you must study in 9th, 10th standard. So these collagen fibers are what? Break off. So they activate whom? Platelets gather. Means they act like a alarm here. Who is act like alarm? Collagen fibers break off from the vessels. They will call the all platelets to gather. Okay. Now, once they gather, there is a form of plug on the cut. So what is the first thing they are doing? It's form a plug. So you can see little blood is not coming out right now. Okay. Just 2 minutes, we will cover this. Okay. Blood is not coming out from that. Here, blood clotting is not done yet. Here, just what? Just preventing of the blood loss. With the help of plug on the cut, with the help of platelet just arrived. Not yet started anything. Then, injured step 1. Thromboplastin forms the enzyme prothrombinase. So who is getting activated after the platelet come into the picture? Here are platelets are inside. Now, once the platelets are arrive on the injured part of the tissue, thromboplastin forms the enzyme called as a prothrombinase, which makes the blood vessel to contract. Why contract? If there is a relaxation, more blood will be fall. That's why if the injury is there, we have to do like this, right? Getting my point? So it catalyzes this. What? And inactivates the heparin. What is heparin? Heparin is responsible for blood not to clot. Heparin is the one who is responsible for not to clot the blood. That's why if the blood taken out from the patient, they add the heparin inside the blood and they store inside. If the blood want to store in the blood banks, the heparin was added. Right now we don't want heparin because we want clot should be formed. So heparin is inactivated in this case. So prothrombin catalyzes what? Prothrombin undergo the along with the calcium inside the blood, thrombin is going to form. From prothrombin in step 2, we got a thrombin. From thrombin, it catalyzes with the help of. So can I say that prothrombin and fibrinogen are the one who undergo simultaneous conversions with the help of blood calcium? Prothrombin will be from thrombin, fibrinogen. The fibers which are getting from fibrinogens are going to form a fibrin monomer. Only single threads we are getting. Yes or no? So this single thread undergo the polymerization along with thrombin and fibrin monomers form the blood corpuscles along the fibrin threads and the blood clot will be formed. So how the blood clot will be formed right now? These are what? Platelets. Now platelets will be formed dendrite-like structure and once they form, what going to happen? They cover it. And here is your blood clot will be there. Is it? If the fresh blood clot is there, I just remove. What going to happen? A pinkish color skin is there. It's about to form. But again, the blood will be coming out. Okay. So this is a mechanism of blood clotting. Okay, just listen. When you talk about the blood clot, I'm not starting that. Blood clot. What is the blood clot? You can have it. What is blood clot? Blood clot is defined as the mass of a coagulated blood. So when you talk about the blood clot is what? Mass of a coagulated blood which contains the RBC, WBC, and the platelets. Okay, so blood clot is what? A mass of coagulated blood. But when you talk about the clot retraction, what is that? Just I want to tell you. Retraction means what? The process involving the contractile of the blood clot oozing of the serum. Means if this is a fresh cut, clotting is there. If I'm just pressing it from this side, what going to happen? A serum going to out that. that called clot retraction. Okay. So contractile proteins, namely actin, myosin, and thrombosin in the cytoplasm of the platelets are responsible for clot retraction. Now, one more point, the last one, which you should know about that, as per the neat perspective. What going to happen if the clot is inside the blood vessel? It is a problem. Are you getting my point? What I'm trying to say that what going to happen if the blood clot, if it is outside, it's okay. But blood clot, if it is inside, what is the function of blood clot? What is the membrane is undergone on? It should be stitched properly, right? So if the blood clot is inside the blood vessel, lumen will get affected. What is lumen? The the inner thing is going to affected, right? Please pay attention here. Just one point. It's called a fibrinolysis. This is very, very important point. Lysis of the blood clot inside the blood vessel is important when the clot is inside the blood vessel. Otherwise, it's going to create a problem for the transfer of the blood, right? What is the function of blood clot? Whatever is the problem is that has to be removed off, right? So it's called the fibrinolysis. It helps to remove the clot from the lumen of the blood. If the lumen is like this, okay, and blood clot is here, what going to happen? The problem, what is going to happen here? Whatever the blood should be passed, it's going to hindered by this, right? Alter by this. So this is to be removed. What is the first priority of function? It will be heal the wound first. Okay. Now once it done, definitely it's get removed. If it is on a skin, it get removed by anyhow, right? But if it is a problem in the blood vessel, it is not getting removed easily. So there is a one more process called fibrinolysis. Clot has to be melt or dissolve inside. So this is called as a fibrinolysis. Let's start. Now lymph is there. During the blood flow through the capillaries, some water soluble substance move out in the space between the cell tissue. Now what is going to happen if this is a network of the capillaries? So what happens? Here is a space. Here will be the little space will be there. Here will be space. Here will be space. Now this capillaries, what is capillaries? They are perforated. What is perforated? They can easily squamous, made up of squamous epithelium, which is a single lining is there. So some material can be oozing out from here to here. Not a form element. RBC platelets are not getting out. Only what getting out? What is getting out? Some water soluble substance, because water can be transferred. Getting my point? Blood is inside. The blood system, whatever circulatory system is a closed system. In a human body, blood system is what? Closed one. So they form from arteries, then capillaries, network of capillaries, veins, again the same, again to the heart, right? Yes or no? So they are a closed system, but there is a space in between. Those are the water soluble substance. They can be coming out from that. It's similar to the blood, but has a fewer blood proteins. So lymph is nothing but what? Filtered blood. Filtered blood from the blood vessel membrane. So what is that? Less calcium, phosphorus, and a high glucose concentration. Why high glucose concentration? Because glucose is water soluble. Even in case of plant, need question will be there. In case of plant also produce glucose, but plant never store as a glucose one. Why? Phloem is there, sorry, xylem is there for water conduction. What happen if the glucose is getting dissolved into the water? पेड़ की तो पूरी मेहनत खराब होगी ना. Whatever the energy has expenditure to form the glucose, if it is not converted to the starch, it will be a great problem to plant only, because glucose is the one who water soluble. So it's better to make it glucose from to the starch. Getting my point? So why we get a starch from plant? Because of this reason. Xylem conducts the water. It may chances whatever the converted factor of the glucose can be dissolved into the water, right? So to prevent that, it's a conversion to starch. Similarly, see, I have given the example of botany in the zoology right now. Okay, so there are similar factors here also. Glucose concentration is more in what? Plasma. Sorry, not plasma. Water, lymph. Sorry. It colorless fluid containing the specialized lymphocytes that provide the response to body. What is exactly lymph? In your nutshell, I will tell you. In blood vessel, there is a WBC, everything is there. They can taken care of the any infection. What happen if this space are empty? If the bacteria is very smart and just he's sitting in this spaces, so who will take care of that? So lymph is going to take care of. So lymph has a lymphocytes that provide the immune response. This point is very clear regarding the lymph. Main function of lymph is to provide the immunity, carry proteins and fats molecules, oxygen, material, thermos. So what is main thing? Immunity. You got the points? If the bacteria sit into the gap where blood vessels are not there, so who is taken care of that part? Lymph will be there. Now, circulatory pathway. Let's move to this part. This is there, right? This, they can't be reduced, this portion. This is has to be there. Circulatory system. What is circulation? Tell me, what is circulation? What is blood circulation? Why there is a blood circulation? क्यों जाता है? What is the reason behind that? Why it's needed? I told you the thalassemia, right? The thing. So what is the circulation over here? Blood is a major connective tissue. It goes to all the body parts, all the cells, gives the oxygen and nutrients like glucose, and taken up what? Waste material that has to become. So circulation has to become like who is the organ called as a pumping organ? Heart will be there. So heart have a two compartment. Majority should be two compartment. One dealing with a deoxygenated blood, one dealing with a oxygenated blood. So circulatory system is a open circulatory system and a closed. We prefer, humans prefer the closed system, not a open system. Why we should not go with the open system? It's found in a arthropod and mollusca. Why? In cockroach, you can see the open blood system. Why? They don't have that complex system. Whatever they eat just has to be stored. Okay. So closed circular system in annelids, chordata. Chordata means what? Those who have vertebrates, right? So we are the one who are from originated from here. So we follow what? Closed circulatory system. All vertebrates have a muscular chamber heart. Okay. Why there is a muscles in the heart system? For pumping mechanism. First, two chamber heart. Fish has only how many? Two chamber hearts. Amphibian and reptile except the crocodile. Three chambers heart is there. So where you find the three chamber hearts? Amphibians and reptile. Amphibians and reptile, mostly the reptiles are a warm blooded or cold blooded? Cold blooded. Okay. So for that purpose, they have need that. Crocodile, birds, mammals have a. Okay, when you talk about the warm blood and cold blooded, once I'm completing this, I'm going to tell you that because after completion this, you will understand that point. So I will tell you what is warm blooded and cold blooded animals and what is the function of that. Okay, let's talk about. Even in the food, I will give the hint. Dahi, like curd. Curd is what? It is good for health, yes, but it is warm or it is cold. Actually, it is a warm. In a case of Ayurveda, what is warm? I will tell you that. It appears like a cold when you're eating it because you keep it in refrigerated, but it acts like a warm inside the body. What is that mechanism? I will tell you. Just first concentrate here, then I will talk about that. Okay, because this mechanism should be there, otherwise you will be forget that. So human blood vessels and the circulatory fluid contains over there. So heart is a mesodermally. Can anybody tell me what is mesodermally? Ectoderm, endoderm, mesoderm. Mesoderm. Zygote is going to form cellular mass. Three division will be there. Mesoderm means middle part. So heart is originated from the mesodermally derived muscular organ present in the thoracic cavity. Thoracic cavity. Sternum is there, right? The ribs are joining here. So lungs are there and the heart will be there in thoracic cavity. I will show you the diagram. Don't worry, everything. Two lungs protected by the double membrane of pericardium. What is pericardium? I told you the example. For example, I'm carrying this two objects, okay, like this. If I just fallen down, this also can spread. But if I'm carrying this object like this, what going to happen? They will not separate it too much. So this is a pericardium, a covering on the organs is called a pericardium. Mostly pericardium is, the cardia means what? Heart related. So pericardia is a covering on the heart. So can I say that here are some vena cava? What is vena cava? Superior vena cava, inferior vena cava. Superior collecting blood from the above, inferior collecting blood from the down. So they are going to pour the blood into the atrium part. Which one? Right atrium. Right atrium is nothing but what? What is it? Atrioventricular node. What is that? SA node and AV node. We're going to talk about it now. Let's talk about this. What is it? Chordae tendineae. These are the fibers. Now this is a flap-like structure. This is a flap-like structure. If one atria got filled, it is going to open and its blood is going to pour in this side. So this is inferior vena cava. This is superior vena cava. They pour into blood into right atrium. Once the right atrium filled up, it goes to the right ventricle. From the right ventricle, from the not aorta, from the pulmonary. Okay, now in the body, in the human body, pulmonary arteries. Pulmonary arteries are the only arteries which carry the deoxygenated blood. And the pulmon, it goes to where? Lungs. And from lungs, oxygen is get added over there. They form the oxygenated. So pulmonary vein will be there. Now pulmonary veins are those veins, only veins, because artery is responsible for carrying oxygenated blood and vein is for deoxygenated. But in this case, opposite is there. Pulmonary arteries are responsible for carrying deoxygenated blood. Please listen very carefully. Deoxygenated blood from the right ventricle towards the lung for getting oxygen to be added. Once oxygen is added, now this oxygenated blood is carried by the pulmonary veins, which are the only veins in the human body which carries the oxygenated blood. Now, once they carry the oxygen blood, they will be poured into where? Left atrium. From the left atrium, there is a, again, not bundle of fish. What is this? Flap is going to open, blood will going to pour here. Okay, and this is the left ventricle. Left ventricle is much more stronger as compared to the other things. Can you see that? Much more stronger because it's the one who pumping the blood all over the body in the form of two via aorta. Getting my point? So primary, this is first, this is second, third, and fourth will be there. I'm going to show you all the three diagrams, but I wanted to do this. The upper two parts of chamber called the atria. One is atrium. It's also called atrium. Atrium means what? Ear-like structure. Atria means ear-like structure. And lower two chambers are called the ventricles. Interstitial septum. What is interstitial septum? This is a septum, which is atria and the ventricles are separated. That's called interstitial septum. Are you getting my point? Everybody are with me, right? Okay, sorry. Correct. The opening between the right atrium and the right ventricle is guarded by the three muscular flaps called a tricuspid wall. So can I say this is a tricuspid? 1, 2, and 3. So can you see there's some fibers are there? These are pulling down. These are pulling down, so blood will be flow from atrium to ventricle. If they're pulling up, what is going to happen? It's a closed. Yes or no? So this is tricuspid wall. What is the wall name as? Tricuspid. Bicuspid or mitral wall guarded the left atrium. Here is a, can you see that only two layers are there? So this is only two part. Here it will be three. I mean, I want to show you like this. 1, 2, and 3. Okay, this is tricuspid, which is found between the right atrium and right ventricle. Only bicuspid is going to found here between the left atrium and the left ventricle will be there. Let's move further. Now, the opening and the right left ventricle to the pulmonary artery and aorta respectively is controlled by the semilunar valve. The nodal tissue present on the upper right corner. What is nodal tissue? See, I will show you that. Now, heart is what? A pumping organ. So there is a, here is a SA node and there is a AV node. Okay, I will going to discuss it later. What is SA node? Which is present here on the septum. This is the one who generates the impulse. So activation will be starting with the SA node. From here, it goes, let me use another pen. From here, the impulse are going here. There is the AV node. And this is a bundle of his are responsible for pumping of the things. Okay, I will show you that. Just understood one thing. What is that? SA node is a starting of impulse. Okay, and it goes to the AV. What is AV? We'll see that. Okay, so let's go further. We just have seen the videos. Now let's start the cardiac cycle. What you're right. To begin with, all four chambers are relaxed state, all called joint diastole. What is joint diastole? Systole and diastole. Systole is the one where the, yes, pumping will be. The diastole will be relaxation phase. So as the bicuspid and tricuspid wall are open, where is you find bicuspid one? Tricuspid is one from the right atrium and to, and bicuspid will be left atria and left ventricle will be. SA node generates the action potential that contracts both atrial systole. Action potential. What is action potential? What is that going on? What is that? Give me a word. impulse, electrical impulse passes to the AV node from SA to the AV. SA is a active pacemaker and AV is the one who transmit it, right? So bundle of his transmit to the ventricle. Who is transmitting to this bundle of his? Okay. Okay. Now semi-lunar valves are there, open into the circulatory system that relaxes the ventricle and close the to prevent the backflow. So in veins, there are some valves are there to prevent the backflow. If there is a backflow, it will be problematic. Why? Proper circulation will be hampered, right? So as the pressure inside the ventricle decreases, the bicuspid and tricuspid valve open to the repeat the process or cardiac cycle. If there is a problem in a valve, what going to happen? Blood filter, I mean not blood pumping will be not properly maintained. Okay, ask me the doubt. Let's talk to here. During the each cardiac cycle, two sounds are produced. In Bollywood, dhak dhak is no sound of the heart. It's a lup dup, not dhak dhak. Okay. So what is lup dup? The first sound is lup is due to the closure of bicuspid valve. So bicuspid valves are closing first, then tricuspid valve. So lup and dup. These are the sound. Okay. Now ECG. Let's go further. We already seen the video. I'm just going move forward. ECG, electrocardiogram, is a graphical representation of electrical activity of the heart during the cardiac cycle. The electrocardiogram machine is used to obtain the electrocardiogram. Okay, you can get this. P wave is the just starting of the SA node. Then this is the relaxation depolarization time. QRS is a shoot up, like SA transferring towards a, okay, contraction will be there. And QRS complex. Okay, now T wave will be the just beginning of the next cycle. So this is a cardiogram. We just have seen in the video as well. Let's go. Now, here we're going to talk about the T wave and what is they are actually. So let's see here. The P wave represent the electrical excitation of atria. That is depolarization. Excitation means what? Depolarization. With what is depolarization? Just start to contraction. Before the contraction, depolarization. That means sodium and potassium ions are or calcium ions are exchanging. Contraction of the atria. QRS wave or QRS complex represent the depolarization ventricle which initiates the ventricular contraction. So QRS is what? Initiation of ventricular contraction will be there. Then T wave represent the return of ventricle. Once the contraction is done, obviously they little take a time. They take a little time to relax, to take origin position, right? So that is called repolarization. So initially D and then repolarization. The end of T wave marks the end of systole. So systolic and diastolic pressure. Systolic means pressure from the high BP and low BP, like systolic pressure and diastolic pressure. Systolic pressure is high and diastolic pressure is less. Systolic is atrial pressure, atria. Okay, we can say the arteries pressure, and diastolic veins pressure will be there. QRS complex in a given period of time determine the heartbeat rate. Okay, now what is double circulation? You are already aware about that. Flow of the same blood twice through the heart, once in oxygenated form and other in a deoxygenated form. So who is responsible for making it oxygenated? Lungs. Who carries that from the right atria? Pulmonary vein, pulmonary arteries, and pulmonary veins carries. Okay, this is a confusion, point of confusion. Please make sure. Now, systematic circulation includes the flow of oxygen blood from the left ventricle to all the body parts and deoxygenated blood from the various body parts to the right atrium. All the systematic circulation start from the aorta at the superior vena cava. I just told you, na, superior vena cava, inferior carry the blood from downside or inside, what you can say, and the superior from above side. The systemic circulation provides oxygen, nutrients. We all are aware about this, right? So this is the double circulation. What is this? Vein and artery. Can you give me differentiation between vein and arteries? Arteries are more in lumen is very less because they have a high pressure. Veins have a less pressure. That's why vein contain the valves. Artery do not contain any valve. Okay. Arteries are mostly under pressure. Veins. Veins, no, arteries. Arteries are the one who carries the oxygen blood, definitely. So they have to see aorta is the one who, when the pumping is done, just done contraction of the ventricle, blood will be flowed through aorta. That is artery. So artery will be under high pressure. That's why lumen is very thick. We can say this thick, but lumen is very small. In this case, lumen is very big as compared to this, and the coverings are less. Okay, thin, you can say that. And capillaries, only the one single layer of capillary epithelium is there. Now, pulmonary circulation. What is pulmonary circulation? If the heart wants to pump, definitely it requires some blood as well, right? So the flow of deoxygenated blood from the right ventricle to the lungs and the return to the, when it going to return to the oxygen blood from the lung to the left atrium is called a pulmonary circulation. Matlab, what is pulmonary circulation? Lungs and the atria, and lungs and the left atria will be there. Okay, so that is okay. Now, regulation of the cardiac activity. This is very important. Okay, I'm covering this chapter little bit quickly. Normal activities of the heart rate are regulated by the nodal tissue, that is SA and AV node, so that heart is myogenic. What is myogenic? The one who can prepare their electrical impulse by itself only. They not require anything. Okay, smooth cells are there. Okay, and myogenic are responsible for what? To carry out their own functions will be there. A special nature, sorry, the special neural central in the medulla oblongata. What is medulla oblongata? What is that? From the brain, where the brain actually extend to the spinal cord, that is the medulla oblongata. So medulla oblongata moderates the cardiac function. If there is a problem in medulla oblongata, problem in the heart rate. Okay. So function by ANS. A sympathetic nerve can increase the rate of heartbeat and parasympathetic way. So adrenal medullal hormones also increase the cardiac output. What is adrenal? Adrenaline, noradrenaline, flight and fight mode. Obviously, we need a more energy, so heartbeat will be more. Now let's talk about the disorder. This is the last part. Let's do it quickly. Hypertension, high blood pressure. Blood pressure higher than 120 is a systolic, 80 is a diastolic. So 120 is a normal thing. If it is more, then what is going to happen? Systolic, that is the pumping pressure, and 80 mg is the diastole, that is the resting pressure. Pressure inside the artery, pressure inside the veins. It leads to the heart disease and affect the vital organ like a brain and kidney. Why brain and kidney? If the blood suddenly shoot up, blood is more in the brain. It will be create a problem. Blood suddenly shoot up, it will be problem to kidney because more filtration will be there. It has to be maintained. Coronary artery disease. What is that? Commonly called a atherosclerosis that affect the blood vessels that supply the blood to the heart. So, suppose this is the blood vessels here. They are giving blood to the heart for a pumping purpose. If there is a clot in between, it will be create a problem for a heart to pump. Okay, that is called coronary artery disease. Angina pectoris is also called. Is also called angina pectoris, acute chest pain. Chest pain will be there due to the less supply of oxygen. Ask me. Ask. Give them. So angina pectoris is what giving the acute chest pain, less supply of the oxygen to the heart muscle. It occurs in the elderly male and female. Okay. Now heart failure, a major issue will be there. So heart does not pump enough blood to meet the required. So angina pectoris is the one who can you have a pain. You can do your activity less. But in case of heart failure, you can't do anything, right? Then it is as known as a congestive heart failure because of the congestion of the lung is it causes. Now heart failures are different from the heart attack. What is heart attack? Heart is not getting proper oxygen, that is heart attack. That's why it's squeezing. For example, if there is a water pipe is going, I'm just doing this. Okay, what going to happen? The moment I remove the leg, what going to happen? A whole amount of blood, like Tom and Jerry में देखा रहेगा ना वो जो पैर रखते तो ऐसा फूट जाता है, right? Similarly, it's going to happen in heart attack. Heart failure is a little slowly process where the cardiac muscle are devoid of their work. Okay, the work is getting lesser and lesser. Cardiac, it's also called a cardiac arrest, where the cardiac muscle is not working as per the up to mark. And the last part, but not the least, that is coronary thrombosis. Coronary arteries are responsible for giving blood to the heart for a pumping thing. If there is a clot, what is going to happen? Formation of clot in the coronary artery is coronary thrombosis. It occurs most frequently in the left anterior descending coronary artery. Why only left? Because majority is the pumping is done over there. Okay, so we have covered this much here.